started here. So first of all, test corrections, they're not due till Monday, but I feel like the way I put them on Moodle might be confusing for one. So because test 2 is on Unit 2's information, I put the test corrections on the Unit 2 part of Moodle. Right? I've had many people ask me that. So just so you know, if you're on going forward, if there's anything related to Unit 2, it's in 2, 3 is in 3, so on. Any questions I can answer before we keep going? All right. I've had a lot of technological issues today with my first class, so hopefully this class is better. I believe we ended on this slide with a quick review of what proteins are used when directly building a DNA strand. So we're going to start here again. Give a nice review of what we learned on Wednesday. Then we'll review and move on into the cell cycle. And if you have any test questions or comments, I'm sure happy to take them. For now, and then once I update you for that, I don't know what Kelly, how is Halloween? Sometimes that's a good thing. Very community trick-or-treaters just due to the weather, I think. So,
yeah, we facilitated that to them much during
oh, okay.
So this weekend is for tonight.
Oh, yeah. That's smart. Who's listening to that? Okay. I work. Perfect Lord Jesus. You have to. Should have got a head start. Or is this your own personal notes? Oh. I'm going to remember now. All right, so there are four proteins involved in directly building a new DNA strand. What might the first one be? DNA polymerase 3. It does a lot of the work. All right, well, I thought they all came up sequentially. DNA polymerase 3 is one of them, right? Primase is another one that I heard. What does primase do? Yeah, it makes an RNA primer so that DNA polymerase knows where to start. What about DNA polymerase 1? What does it do? Yeah, we have to get rid of those RNA primers. So this replaces the RNA with the DNA. And then finally we have ligase. What is the purpose of ligase? Connects it all together. Yes, makes it one nice new strand. All right. So we've learned a lot about DNA so far, and we're not done yet. So we learned the structure of DNA. So how we go from a nucleotide to a strand to a double helix to a chromosome. All the chromosomes are the genome. We know how DNA replicates, and we know that DNA replicates when a cell is about to divide. That leads us to our next topic, which is cell division. So chromosomes, they play a key role in cell division. Right? We want to make sure that each resulting cell has the right number of chromosomes, right amount of genetic material. When chromosomes get ready to divide, remember how we talked about levels of compaction in a chromosome? And how euchromatin was compact, but then heterochromatin was even more compact? Well, it's during cell division that they become this heterochromatin, a super condensed version of the chromosomes, so much so that we can often see it just under a light microscope. Right? We can see individual chromosomes. If you're into this type of field of study, this is cytogenetics, right, where you're looking at genetics by looking at microscopic work of chromosomes. I know a lot of plant scientists, because I'm a plant person, a lot of plant scientists will do this to see how many chromosomes a plant might have. Plants are weird. They have odd numbers of chromosomes. All right, so chromosomes. We've learned a lot of terminology so far, but we have to understand just a few more bits in order to understand cell division. All right, so we're going to turn our attention to this thing called ploidy. So ploidy is the number of each type of chromosome. So remember, we have a variety of chromosomes. Like each chromosome carries a specific unit of genetic information. Right, so maybe one chromosome has hair eye color, another chromosome has, your shoe size or how tall you're going to be. Right? So ploidy refers to the number of chromosomes for each of that type of genetic information. If you are a haploid organism, you only have one copy of a chromosome. You do not have any homologous pairs. You only have one copy. So if you look at this individual up top, right, it only has 3 chromosomes in total. Right, so the genetic information is spread across the 3, but there's only one copy. Right? So only one red, one purple, one yellow. Versus diploid organisms, which we are predominantly diploid. Right? A lot of organisms are predominantly diploid. They have pairs of chromosomes. Right? We call these homologous pairs. Right? 2 copies of the same genetic information, but each pair comes from a different parent. Maybe contains a slightly different variation on the genetic information. So blue eyes versus brown eyes. So this individual here, right, it has 6 chromosomes in total, but we have 3 homologous pairs. This is a haploid organism. Now we can go beyond diploid and haploid. Some organisms are triploid, which means they have 3 of each type of chromosome. So instead of now saying they have a homologous pair, we would say they have a homologous triplet, right? Because there's 3 of each type of chromosome. That's what this bottom image is showing you here. Right? Each type of chromosome has 3 copies. We can have ploidy that is a tetrad, right? Or it could be have 8 copies. There could be one, I don't even know the word for it, but they could have 10 copies of a particular chromosome. Right? Lots of times plants have those odd numbers of chromosomes, where they might have 10 copies of the same chromosome. In humans, what we're mostly referring to, unless there is a genetic abnormality, is diploid or haploid. Right? Either you have 1 copy or you have 2 copies of your chromosomes in your cells. Make sure not only do you remember haploid versus diploid, but take note of the shorthand way that it's written out. So haploid is an N, diploid is 2N, right? Triploid would be 3N, and so on. we'll see the shorthand version come up quite a bit. All right, before we move off of terminology, I mentioned this on Wednesday, but I want to make sure we have it down, right? That chromosomes, most of our chromosomes are called autosomes. They contain the bulk of our genetic information. So in humans, we have 23 pairs of chromosomes. 22 of those pairs are autosomes. Tell us hair color, eye color, heights, what proteins to make, when to make those proteins, all of that information. That last pair would be our sex chromosomes. So XX if you're a female, XY if you're a male. Right? The sex chromosomes contains all the genetic information into making you either male or female. Right, so all organisms kind of have this divide between having autosomes and sex chromosomes. If you combine them all together, this is kind of a side note, I wouldn't expect you to know this term, but up here we have two images of different genomes. Top one is the human genome, because we see it as 23 pairs. All of the chromosomes lined up together, right? Notice how they're split into homologous pairs. Right, so 1, 2, and 3, 1 is all the same size, has the same type of markings, same with 2, same with 3. Right? When we put them all together, we call this a karyotype. So a karyotype is just the full visual image revealing the number, the size, and any characteristics of the chromosome. Now this one down here, I forget what genome it is. Maybe I put it in the notes of the PowerPoint slide, but it's definitely not human. Because if we look, they have 40 pairs of chromosomes. Right? So, number varies depending on species. All right, so let's do another review here. So first of all, we're going to review the terminology about chromosomes because, again, it can be complicated. A lot of terms sound the same, and we're going to be using these terms quite often. And then I want you to determine the ploidy level for these two groups of chromosomes. And let's see, I don't have a timer on this slide. We'll do about 2 minutes. That's the homologous. all these chromosomes. Let's start with A, which might be the most tricky one. What is A in this image? It's just a chromosome. Yes. So A refers to this as a whole, both sides of it, right? But that structure as a whole. So A is the chromosome. So in this image, we have two chromosomes, right? One is this blue, one is this pink. All right, what is B? Centromere. And C? Not a homologous pair, but sister chromatids. So remember, sister chromatids, they're identical. They're on the same chromosome, right? Either side is identical to each other. So if C is a sister chromatid, what does that make D? The homologous pair, yes. It's the same type of genetic information, but a variation on it, right? So think, I like to use hair color and eye color. Maybe blue eyes versus green eyes, right? Codes for eye color, but different variations of it. All right, these sets of chromosomes at the bottom. What is this first set? Is this a, or what's the ploidy level of this one? Yeah, it's 2N. It's a diploid, right? Because we have 3 sets of chromosomes, right? But within the sets, we have 2 copies carrying that same information. So in this image, they're color-coded. Those homologous pairs are color-coded. All right, if this one's 2N, what does that make the other one? 3N. Yeah, and instead of saying we have a homologous pair, because we have more than 2, we would say we just have homologous chromosomes. And then you would say a homologous triploid, right? Because we have 3 containing the same genetic information. Any questions before we move on? All right, so let's talk about cell division. Right? Cells don't just arise from nothing, right? They have to come from something. So new cells arise from pre-existing cells. And there's 2 ways that a cell could go through cell division. They might either go through mitosis or meiosis. Meiosis, we have a diploid cell if we're human, creating another genetically identical diploid cell, so 2N to 2N. In meiosis, we're starting with a diploid cell, and we're ending up with a haploid cell. So we end up with half the amount of genetic information. Okay, so we go from diploid 2N to haploid, which is 1N. And we're going to go through both of these in depth here. We'll start with mitosis today, and maybe finish mitosis, though I feel like we'll run out of time with the content check. But both of these, the last step is cytokinesis. And cytokinesis is the process of dividing the cytoplasm in half in a cell that's going through division. So here we have a cell. It's almost completed cell division. Right? You can see it has 2 sets of DNA, right? 2 nucleuses. Right? We want to split that one giant cell with 2 nuclei into 2. And we're going to do this by splitting the cytoplasm through cytokinesis. So essentially we're pinching them together until they separate into 2. All right, so mitosis versus meiosis. Like I said, we're going to focus on mitosis today. That means we're creating genetically identical cells. We look at a little bit further what the difference is. I just really want to reiterate the difference between meiosis and mitosis. Meiosis leads to the production of gametes. Gametes, they are haploid. Those would be like egg and sperm. Or if we're talking about other organisms like spores of plants. So there's gametes. Half the amount of genetic material as the parent. Right? So instead of having 2 copies of each chromosome and being diploid, gametes are only haploid. They have 1 copy. Mitosis, on the other hand, they produce all the other types of cells in our bodies, which we call somatic cells. So anything that is not a gamete is a somatic cell. In mitosis, we're going from a diploid cell to a diploid cell. Right? The cells being made are going to be genetically identical. Right? So that genetic material is copied and divided equally. We can see that in this image here. Right? Homologous pairs, they're color-coded. In this diploid somatic cell, right, we see we have homologous pairs, the oranges and the greens. Right? Diploid cell versus a gamete that is haploid. We only have one copy of each. Meiosis, production of gametes, haploid genetic material. Mitosis, production of somatic cells, genetically identical, same amount of genetic material. All right, like I said, we're focusing on mitosis, which means we're going to focus on where we go from a diploid cell to a diploid cell. Daughter cells are identical to the parent. Now, there's a few reasons why cells might go through mitosis, right? I'm going to continue on, but I'll answer your questions on the next content, or the next discussion. How about that? All right. So mitosis, we're going to keep making identical cells. So mitosis, you have single-celled organisms that might undergo this. Right? So if you're just a single cell, you don't need to make another cell to add to your body, right? But instead, you're making another cell as another individual. So this would be asexual reproduction, right? You're creating a new individual with the same genetic information as the original, but it's a completely different individual. Not just another cell in your body. Multicellular organisms can also go through asexual reproduction. Right? So for example, this is a hydra species in the bottom corner. Right? They are a marine species. They live in the oceans. What hydra will do is basically start creating another individual off of its main body. And you can see that here. It's kind of budding off. Eventually, this will completely dissociate from the main body and become its own individual. Now, they're genetically identical. They're basically clones of each other. Right? Same process as these single-celled organisms, just now they're multicellular. But beyond asexual reproduction, we might go through mitosis as a form of growth. Right? So think of how much bigger you are now than when you were a toddler. Right? You've added on to your body mass, right? You have more cells in your body, right? So as you grow, you're going to add in more cells. These are all going to be somatic cells. But also like plants, right? Plants starting as a seedling and growing, producing longer stems, more leaves, bigger leaves, right? They're adding to their body mass. You need to multiply the number of cells. But also repair. Repair is a big one. Broken bones, or you fall and scrape your knee, right? You need to replace maybe cells that were lost or cells that have been damaged in this process. You need to repair your body, right? This is another reason why we might go through mitosis. All right, so let's stop and do a review. Even though you don't know all the steps of mitosis and meiosis, you should be able to tell me based on the visual clues which one is mitosis, which one is meiosis. All right, so if I were to look at these two diagrams, let's look at the first one here, the one on my left. What does this represent? Mitosis or meiosis? Mitosis. Why? What cued you into this? Yeah, it goes from 2N to 2N, right? The parent cell is genetically identical to the daughter cell. Right? So if this one's mitosis, we know the other one has to be meiosis. And again, we can look at the ploidy levels here. The parent cell versus the daughter cells. Daughter cell only has half the amount of genetic material. Right? They're only a haploid versus the parent was a diploid. All right. These next few slides, I promise I have tried to make them as exciting as possible, but it's not the most exciting information. It's a lot of terminology. So a cell is going to spend all of its life in what we call a cell cycle. Right? Think of it as like the lifespan of a cell. Right? It lives in this cell cycle. So it's a series of events that results in cell division for many cells, right? And it's very highly regulated. Right? Because these cells, as they go through their lifespan, right, they're eventually going to divide. We want to make sure we regulate when it divides and to make sure it divides accurately. All right, so the cell cycle, many stages. Okay, we have the M stage, the G1 stage, the G0 stage, which is kind of like this little side bubble here. We have the S phase and the G2 phase. We're going to define all of these. Yes. Yay, I like the enthusiasm. All right, we're just going to start by defining the mitosis stage from interphase. Right? So if we look at the stages, we often start at G1. This is called the first gap phase. Right? The second phase is the S phase. This is specifically where we're synthesizing DNA. So S phase synthesis. Right? And then we have this G2 stage. These three stages together are called interphase. Right? So sometimes you'll hear that during interphase, this cell did X, Y, and Z. Right? Interphase is the combination of these three stages. The next big part of the cell cycle would be the mitotic phase, the M phase. This is where a cell will divide. It will go through mitosis and cytokinesis. Now, some cells may never divide, or they might postpone division. And when they do this, they go on this G0 phase. Kind of this smaller bubble off to the side here. They just kind of hang out. So if they're not dividing or never going to divide, they're in this G0 phase. So for example, a lot of mature neurons, they will never divide. they're always going to be in this G0 stage. All right, so we have all these stages. We can broadly split them into 3 categories: interphase and mitosis, and then this G0 phase if we are postponing or never divided. Let's take a closer look at interphase. Right, so interphase, we have first gap, the synthesis of DNA, and the second gap. Well, I have told you what happens during the S gap, but I haven't told you what happens in the other 2 stages. So in interphase here, the first gap, what the cell is doing, it's growing. It's getting ready to divide, right? It's growing bigger. It's starting to accumulate any molecules it might need for division. Organelles are starting to divide. Right? It's getting ready for cell division. Once it's grown to a certain extent, it'll move on to that S phase, which is where it's synthesizing DNA. This is where DNA replication takes place. Right, so when we talk about DNA replication, I often said it happens when a cell is getting ready to divide. This is that stage, the S stage. The last part of interphase is the G2 stage, called the second gap. Okay? And what the cell is doing here is it's just continuing to prepare to divide. It's growing even bigger, so there's more growth happening, and it's accumulating any proteins it might need during cell division. All right, so interphase, 3 stages: G1, S, G2. G1 is cell growth. S is DNA replication, or synthesis of DNA. G2, more growth. Right, so there's growth, synthesis of DNA, growth. Remember, during synthesis of DNA, we're not just replicating one chromosome or half the chromosomes, we're replicating every chromosome in the cell. This is where we go from a single looking chromosome to an X-shaped looking chromosome. This is where our sister chromatids are added. All right, so I mentioned we have interphase, but a cell can also hang out in that G0 phase. Okay? That G0 phase is where it's not divided. So what leads a cell to divide? A couple of things. One is external factors. Right? So we have to make sure that the cell has enough nutrients around it. Right? Or maybe the temperature is just right. Maybe the pH levels need to be right. Think about plants. Right? Plants don't grow if they don't have a lot of sunlight. Plants have to have an external environment of lots of sunshine so they can photosynthesize, have the necessary nutrients to create more cells. Right? So anything exterior to the cell would be its external environment. Cells can also signal one another when it's time to divide. I'm not going to focus too much on that, simply because we haven't learned cell signaling. That if you're interested in it, you will learn that in a future class. But then there's these internal factors, factors within the cell cycle itself. Right? So the lifespan of our cell. Within the lifespan of the cell, there's different molecules that will help regulate when a cell can move forward into the next phase. Right? We call these the checkpoints. So different checkpoints scattered about in this cell cycle. Now we're going to talk about 3 different checkpoints. So those 3 checkpoints, they occur at different parts of the phases. The first one would be the G1 checkpoint. So in the G1 checkpoint, we're basically checking for, do we have enough nutrients to continue growth? Right? Do we have enough growth factors, which are different molecules needed for division and growth? Is there any damage to our DNA? We don't want to continue this cell into the next phase of cell division if the DNA is damaged. Right, so we have this first checkpoint in G1. Once it passes the checkpoint, right, it's good to go. It has the green light. It's going to go into S phase, duplicate its DNA, and then go into that G2 phase. At G2 phase, we have our second checkpoint. Right? At this checkpoint, we're making sure that the cell has grown appropriately. Right? So we're making sure the cell size is appropriate. We're also making sure that replication has indeed occurred and occurred accurately. Because again, we want to make sure this cell is healthy, ready to move on to the next stage. We don't want to be duplicating a cell that never went through replication, or the replication was had errors in it. Right? Once we pass this checkpoint, then the cell will actively divide during mitosis. And in mitosis, there's yet again another checkpoint, right, specifically at the metaphase part of mitosis, and we'll talk about this in the next few slides. But it's checking to make sure that the chromosomes have been properly attached to what's called the spindle apparatus that will eventually pull them apart. Okay? So 3 different checkpoints. All right, so we want to make sure that cell is healthy, is ready, is not damaged before it moves on to the next stage. That way we are replicating healthy cells. If for some reason it doesn't pass a checkpoint, either cell division is going to be delayed until it can be remedied. That's the word I'm looking for, right? So maybe the cell hasn't grown enough. So it's just going to pause there until it has reached the appropriate size. But it might not even get past a checkpoint at all. It might hit a checkpoint and then just be stopped, right? It's never going to move on to the next phase. Maybe the DNA is very damaged, right? Maybe replication never happened of the DNA, right? That cell is just not going to move forward. Now these checkpoints, they are extremely important to the point where if an individual has mutations where these checkpoints are no longer regulating the cell cycle, this can lead to accumulations of lots of mutations or even cancers. Right? So these checkpoints are very crucial in regulating cell division. All right, I believe my next slide here is a review. Yes, it is. So I want you to review just what are those 5 stages of a cell cycle, what happens in each. right, if we look at the cell cycle, what is the first stage? G1, what happens in G1? Cell growth. If the cell is not going to divide, it's going to move on to this next stage, which we would call G0. It's just kind of hanging out. Okay? But if it is going to divide, it'll go from the G1 stage to which stage? S phase, which, what happens here? DNA synthesis. And to our last stage, G2, and what happens here? More growth. Yes.
That was
Is that 4? Oh, I forgot. But what did I forget? Mitosis, the S phase. Yes, thank you. Thank you for keeping me in check. All right, so mitosis is that last phase where we are actively divided. And mitosis, M phase, is where we're going to continue for the next 5-ish minutes before we get to the content check. All right, so M phase. This is where mitosis occurs. Right. So we are taking the original cell, which we would call the mother cell, and producing daughter cells, which are those new cells. Right. The cell divides, we're creating a genetic copy, and in the process of mitosis, we are going through a series of phases. Prophase, prometaphase, metaphase, anaphase, telophase. Right. We will define each of these out. And then at the end of all these phases, right, we're going to have cytokinesis. We have to divide that cytoplasm in half in order to pinch apart one large cell into 2 new cells. All right, so we're just going to jump right on into these phases. We're starting with prophase. And I'm going to try to draw this out as we go because I think it helps me slow down for you to take notes as well. So, in a prophase, right, we know the chromosomes have already been duplicated during the S phase and interphase. But in prophase, those chromosomes are going to become highly condensed. You know, chromosomes, as they exist, are pretty condensed in themselves. Right now, we're making them super condensed. So chromosomes are condensing. The nuclear envelope, right, how the DNA is held in the nucleus. The membrane surrounding the nucleus is starting to disappear. Right? It's starting to go away. So I'm going to draw my cell. I'm going to do two homologous pairs. My nuclear envelope is starting to disintegrate. And the last thing that happens in this phase is the mitotic spindle is starting to form. We don't talk much about the mitotic spindle. In this image, it kind of looks like, because you can put in my mind, it looks like these spiders around the outside of the nucleus there. Right? So I'm going to draw in my mitotic spindle. Starting to form. Right? Notice it can, as like the center part, and then these fibers radiating outward. All right, prophase. Chromosomes have condensed. Nuclear envelope is starting to disappear, and then our mitotic spindle is forming. Let's just take a closer look at the mitotic spindle because it plays an important role in organizing the chromosomes. You can kind of think of it mitotic spindle as like the puppet strings that are controlling the chromosomes. Right, so they are composed of microtubules, which are these long, skinny strands radiating out. And they're radiating out from the center point, kind of this control center that we would call a centrosome. And now the centrosome sounds really similar to centromere, which is kind of that midpoint on the chromosome. Make sure to keep those straight. Okay, but we have a centrosome, and radiating out from that are those microtubules with mitotic spindles. Now, normally a cell only has one centrosome. If there's an interphase, the centrosomes have also duplicated. That way there are 2. But we want 2 because we're going to end up with more phase here. So from prophase, we have this in-between stage, prometaphase. Now, some textbooks actually don't even mention prometaphase. I like that. I like mentioning it because I think it provides a complete picture of what is happening between prophase and between metaphase. Right? So it's prometaphase. Then we have our cell. We have our chromosomes. At this point, our nuclear envelope is completely gone. Right? There is no more nuclear envelope. Our centrosomes have moved apart and are now at the poles. So notice in my first drawing, they were not completely opposite from each other. Now in the second drawing, I'm going to make sure they are opposite. And the mitotic spindle is now going to be attached to the proteins. So they weren't attached to prophase, but now they are attached in prometaphase. And what happens is each mitotic spindle will attach to each chromosome, but to different sister chromatids. And so if we look at our chromosome with two sister chromatids, in the middle we have the centromere. And during replication or during this attachment, it's going to attach to this chromatic point, which is kind of like a mediary in-between spot that connects this mitotic spindle to the centromere itself. Right? So it's connected to either side of each of the chromosomes. Alright, so essentially, if our chromosomes were the puppets, we are now attached to puppet strings. We're ready to move these chromosomes about in the cell. Alright, I want to give you enough time for the content check. So we will continue with the next phase, which is metaphase, on Monday. All right, exam corrections due Monday. Content check number 6 is up. It is not due till Wednesday, but you can get started on it. But the content check number 5, it is a drawing one. Right? I am a firm believer in drawing things out that helps you memorize them. I want you to draw a DNA replication bubble and include the following terms: leading and lagging strand, template strand, DNA polymerase 3, helicase, Okazaki fragments, and then label the directionality, those 3' and 5' ends. Once you're done, right, if you are hand drawing it out and not coming in digitally, you can make pile up from. You are free to go. Enjoy the evening. Lots of questions. All right. Replication code, that's where the point would be. Yeah. Oh, the gap itself is called the problem. Okay. Almost. Just so like using this, I'm not going to drop my names. I'm not having to draw polymerase 1 or 5 days. It's like a snapshot of it. Oh, it's about that.
Yes, because I probably won't.
It looks like it's open.
There were still people on there. If you need someone,
Everybody know how to make the bubble?
Yes. So it feels like it's in the teens, templates for that. I do that all the time, especially with the personality.
And then the one
Bill was wine, Bill got in with wine. So, what is a template? Yeah, it's the original strands. It's providing the template or the instructions to build. So like leading and lagging are both template strands. Leading and lagging indicate directionality of the template strands. I just had to stop by the I can read. I'm leaving end of
All I do is a 10%.
As long as you're like not doing it to be rude about it, I don't care. Personality. What?
What causes support? Right? And the bubble causes support. The force is
we're good. So it's Netflix and oh, so it's that. So basically, it's like template of the lighting.
Did you guys like the last stream?
Oh, wait, did you guys like the last stream?
It was the easiest laugh. Oh, I think so. On the inside and outside as well.
Yeah, personalities are just really important. Yeah, I remember it's red to get by, milk by the
Somebody there. Oh, is that what the traffic light?
exactly.
I didn't turn my test corrections yet, but right here.
Yeah, I have another pile.
I have a question.
Yeah.
If I have,
Why would you
if
wait
I
to
got
turn?
0.5, like 1 off of the question, but it won't cut back to 0.2.
Guess it depends on the question.
Okay.
One of them you get
One of
full
them,
points.
I didn't even want me to put next in. It's just right there. I just kind of explained it wrong. Here's the number. So I just completed it. So the other
You get 0.25 after that one.
okay.
Looks like half a percentage.
Yeah.
Oh, I guess I'll.
My other, I turned the rest of
Or content test exam. Ones I know who would do.
I didn't send my video.
Hey, what did this do?
Content test?
Yeah.
What a class. End of class, lab, Moodle, I have such a good one.
Okay,
Maybe
so
like an hour
bad.
buffer.
Transferring.
Yeah, that's why you get buffer, so you have some. Honestly, I'm not ready today, so.
I didn't hear from you. Thank you. Walk about a little You're an inspiration. Thank you.
Yeah, we can.
You too.
You can put it on the other pile. There we go.
Have a good weekend.
Yeah, you as well.